Okay, hello everybody, welcome back. This is the second video that talks about speciation. We've talked about how natural selection plays a role in how organisms change over time. We've talked about how isolation is the key to causing one population, one group of individuals that are the same, to diverge or to change into two different types of species. One factor that we started to discuss at the end of the last video is something that is referred to as genetic drift. So when the genes are not flowing from one population to another anymore, we start to see some mutations build up in those isolated populations. With genetic drift, there were two types. One was referred to as a bottleneck, and the other one is referred to as the founder effect. Now, the overall picture is that both of these two types of events take one population and completely reduce those numbers quite a bit, all right, to a very small population size. And then it's those small population sizes that are responsible for making that population big again, okay? Now, the question is, how does that population size get reduced? All right, and that's what brings in the bottleneck, which I just yeah, that's right, bottleneck, and the founder effect. So with the bottleneck effect, we'll go over both of these, and then I'll give you some examples. So again, the bottleneck effect is when a catastrophic event occurs. All right, we've got the beads here again. A catastrophic event occurs that's going to drastically decrease the number of individuals in that population. And so your surviving population doesn't necessarily have all of the alleles or all of the colors that were true of the original population. Okay? Selection cannot increase the frequency of the green gene, or in this case the yellow gene, because it's not there anymore at the end population for us to worry about. So we will no longer see that yellow gene. Okay? Even if that yellow gene at the time happened to be the best gene that was available. Okay, so now that it's gone, it's gone. It's completely out of the gene pool and we cannot get it back. Okay, now maybe a mutation later on in the future will happen and it will bring that yellow back, but for now what we see is there's no yellow gene, and so there's no chance that it will come back. Okay. Selection, remember, selection can only act on what variation is already in a population. It cannot create that variation. Okay, so what I'm going to do over the next few slides here is I'm going to give you some examples of things that have happened in real life of the bottleneck effect. You might want to just write down the fact that the northern elephant seals went through a bottleneck. You don't have to know all the specifics. You do need to know that they were hunted to almost extinction, down to maybe as few as 20, and then those 20 kind of rebounded. So don't get carried away with trying to write down every single thing on the slide. Just kind of get the big picture here. Okay? All right, so the northern elephant seals is an example of a bottleneck effect. It took place between the 17 and 1800s, so that's about a 200-year span. But because they were hunted so heavily, we took a huge population that stretched on the western coast down to about 20 individuals, as I stated. Now, in the early 20th century then, Mexico actually went through and started to preserve put a refuge down and started to preserve these elephant seals or protect them. And then the U.S. government stepped in and helped along the way. And so this diagram down here shows where the original reservation or preservation area was of the elephant seals, and the light blue shows that of today where the population is found. So you can see that the numbers have increased greatly. Okay, I think we're up to about 30,000 individuals now, and so they spread all along the coast of the Pacific Ocean. Okay, now one of the things that we see though is that because we were started with only 20 individuals and it's only been 100 years, so we're not talking about a lot of generations and not a lot of time for mutations to take place, that those 20 individuals that were left to repopulate inbred and mated with one another and spread that population. And because they did that, their genes are virtually all the same, okay, throughout all 30,000 of the elephant seals. Okay, now that's catastrophic when we talk about populations. We like that variation. Remember, natural selection can work on variation. So if everything has the same genes, we worry about if a disease were to come in, because remember, we talked about HIV. Not everyone's going to die if HIV goes airborne, because some people have that variation that will protect them from dying or contracting HIV. All right. Now, if we take a look at these elephant seals, if they all have the exact same genes and a disease comes in that wipes out one, because there's a lot of them now, that gene or that disease is going to spread very quickly through the entire population and could catastrophically cause these guys to go extinct. And so that's one of the concerns they have about these bottleneck effects, is that as they rebound, they get genetically identical and we start to have problems. All right, we also see a similar situation with the cheetahs. There were 4 different subspecies of cheetahs that once roamed North America, Asia, Europe, and Africa. But again, due to hunting, we took the population and we decreased them drastically. Okay, and so now we've spent some time putting them on wildlife preserves over in Africa. That's the only place that they are found, and they have been inbred so much that, again, these animals are also genetically identical to one another. They are genetically identical so much that they can take skin from one cheetah and graft them or place them onto another cheetah, and the new cheetah will not reject that tissue as being foreign. Okay, now let's put that into you. If we were to take somebody else's tissue, whether it's skin, whether it's a heart, whether it's organs, remember all those cells have marker proteins on them that determine who they belong to. All right, and so if you took mine, I'm Mrs. Warren's cells, and you put my skin or my heart into somebody else, we'll say, let's put it into Alex. Okay, so we're going to take my skin or my heart and we're going to put it into Alex. Alex's body then is going to say, whoa, that's not mine, and it's going to reject it, or the immune system is going to attack it, and it's actually going to not only kill the organ, but will also kill the person that it is. So Alex, you would not be here. I'm sorry. Okay. So with the cheetahs, they're so identical that every cheetah recognizes every other cheetah's cells as their own. So again, if a disease comes in, they're worried that it's going to decimate all of the cheetahs. Okay. All right, now we move on to the founder's effect. Okay. Oh, let me go back one slide here. All right, so with the cheetahs and the northern elephant seals, those are both due to bottleneck effects. We had large populations and hunting. It was fairly quick. Okay, 200 years doesn't sound quick, but it's pretty quick. And in case of the cheetahs, it was only about 20 years. But we went through and we drastically cut those populations in half. And we actually today see the same thing happening with the elephants in Africa. Okay, so now we've got the founder's effect. Okay, the founder's effect, remember, is a small group of organisms that move to a new area and take up residence. So now we see the founder effect. And the founder effect, remember, is a small group of individuals from a population that move to a new area and they find somewhere new to live. All right. Now, these guys are reproductively isolated. They're not mating with the major or the numbers of individuals from that original population. So we see this with the Amish culture. A small group of German immigrants came in about the mid-1700s. And with that small group, some of the individuals in there had a form of dwarfism that included polydactyly. Now remember, polydactyly is that characteristic that gives you 6 fingers and 6 toes. All right, so mixed with dwarfism, it's pretty rare in the general population. It's something like one in like 5,000 individuals or something like that have that in the general population. But because we had a small number of individuals that came, the original people, King Samuel and his wife, started this Amish culture, and they are reproductively isolated because they are not mating with everyone else. Now, we may talk to them, they may socialize a little bit with all the other people, but they are not mating. And so that gene that was found within this small group stays within this small group and becomes more and more and more common. So that today we see it as 1 in 7 Amish people have this characteristic. Okay, that's pretty high. In Montana, we also have another example of a founder's effect, and that happens to be with the Hutterite colonies. Again, we see them all the time in town. They're shopping, they're participating in life, they're doing all this stuff. We can go to their farms, we see them at the farmer's market, but they also are reproductively isolated from the rest of the culture. And so some of the stuff that we see with the Hutterites is that they've got this gene for horrible eyesights. And if you've ever talked to some of them up close, they've got those super, super thick glasses. Some of them are virtually blind. Not blind, but they need the glasses to be able to see properly. And so they're doing a number of things to try to kind of, what we say, thin out the gene pool, bring some fresh blood in so that they can try to get that gene diluted, trying to get it to kind of go away a little bit. They will take some of the people from our Hutterite colonies and send them up to Canada. They'll move Canadians down to Montana. They'll send them over to Idaho and some of the other places as well. So they try to get them to mix around a little bit instead of just staying within their own colonies themselves. Okay, so we've talked about two types of genetic drift. Okay, and so one of them is the founder effect, where we have new individuals finding a new spot to live. And again, as these mutations start to creep up in either population, those two separate populations now will become different. And if they become different enough, then they are considered two separate species. Now, does that mean the Amish are separate species than the rest of America? No, because there are instances where people leave the Amish culture. There are people who leave the Hutterite culture and mate with the general population, and we are still making babies, and the babies are still making babies, and life is great. We are still the same species, all right? But they are genetically different in some aspects, or just make them more common. But if this continues long enough for a certain species, they will become separate species, okay? Bottleneck, same thing. We will reduce that population by a catastrophic effect over time. This could result in some drastic differences. Okay, and I hope you've learned a lot about how we can become separate species. Isolation is the key. Isolation between two separate populations.